So here you are, and if you remember in the previous lecture, we discussed the simplest type of plasmid vectors, that is PBr322. And in PBr322, you have all the basic features of a plasmid, which includes the origin of replication coming from Escherichia coli. And then you have two selection markers. One is the ampicillin resistant marker, and the second one is the tetracycline resistant marker. And usually you would insert your DNA of interest into either the tetracycline resistant marker or the ampicillin resistant marker. Let's say, for example, you insert your DNA of interest in the tetracycline resistant marker along one of these restriction sites. Then the way of screening for recombinant transformants is via replica plating. If you remember, we had done this in details in the last lecture. So here you are. What you do is you make a plate first of ampicillin and plate your transformation mix here, and you would have all transformants growing up here, whether they have the recombinant plasmid or the self-ligated plasmid. And then from this, you make a second plate via replica plating, and in this plate, you add both ampicillin and tetracycline. So in the cells where the DNA of interest is present, that is the recombinant colonies, the tetracycline resistance would be lost, and therefore they would not grow here. You can then compare these two plates together, and the colonies not present in the ampicillin and tetracycline plate as compared to the ampicillin plate alone are the ones that represent the recombinant colonies. And so this is a two-step plating procedure, and it is a bit complicated, and therefore what people wanted was a slightly better one-step method of identifying the recombinant plasmids. And that is when you started with the blue-white screening method of plasmid selection. And that is where the Parch vectors come in. The Parch vectors are slightly advanced plasmid vectors as compared to PBr322, in the sense that they have what is known as a blue-white screening procedure. So here is a Parch vector for you. Parch vector, you have one resistance marker to differentiate between transformants and non-transformants. And within the transformant category, you could have self-ligated plasmid or a recombinant plasmid. To differentiate between self-ligated plasmid and a recombinant plasmid, you have the lag Z prime alpha locus here. What happens is that basically you're looking for expression of beta-galactosidase. The omega fragment of beta-galactosidase is expressed from the host cell, while the alpha fragment of the beta-galactosidase is expressed from the plasmid. And in case the plasmid has your DNA of interest, because the MCS is within the lag Z alpha locus here, then in that case, the alpha fragment of beta-galactosidase is not formed, as happens here in this case. What beta-galactosidase does is to basically cause a change in color in the presence of X-gal. The X-gal is converted into a blue-colored product, and therefore the non-recombinant colonies are blue in color, whereas the recombinant colonies where the gene of interest has got inserted into the MCS and therefore disrupted the lag Z prime alpha locus, and therefore the alpha fragment is not formed, and hence functional beta-galactosidase is not formed. In such cases, the X-gal is not converted to blue color, and therefore what you get is actually a white colony. So this is a one-step screening for recombinant plasmids here, and that's the advantage that you have with Parch vectors as compared to the normal PBr322 vector. And as an activity, I ask you a simple question. Enlist the two major limitations of plasmid vectors. Before moving on to Parch vectors, let's also do a bit of revision questions. So here is the question number 1. Question is simple. In blue-white screening, the recombinant colonies are, and I'm sure all of you would be able to answer this. I'll give you 10 seconds to answer this. So your time starts now. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1, 0. The answer for this would be white, and I'm sure everybody would have answered it correctly. So this is white in color. The recombinant colonies are white because functional beta-galactosidase is not formed here. In comparison, the non-recombinant colonies where the plasmid self-ligates and is able to synthesize the alpha fragment, which then couples with the omega fragment and forms a functional beta-galactosidase, the colonies turn blue in color because of the conversion of X-gal to its blue-colored product. Right. Let's do another question here. So here is your second question. In blue-white screening, which parts of the lag Z protein are coded by the plasmid and the host cell, respectively? So here in this case, as we've discussed in the previous question, the alpha fragment is encoded by the plasmid and the omega fragment is encoded by the host cell. So A is the correct option here. And I'm sure all of you would have got it right. So before we move on to Parch vectors, let's do one more question on the plasmids here. So here is a question again. Name the enzyme that facilitates blue-white screening. And I'm sure all of you would get it right again. This is beta-galactosidase. So with those questions as part of the revision strategy, we now move on to Parch vectors. Right? And in Parch vectors, we're going to deal with two different types of Parch vectors. The first and the common one is lambda Parch. And then we also move on and discuss a bit about M13 Parch vectors.